NANI?!
Yo, and welcome to Shooting the Soil. We're doing microscopy May. This is the last one we're gonna do. I might do one more June one, depending on if you guys show some excitement for it. But we're definitely gonna be doing more microscopy. Um, but anyway, yeah, we shoot the soil with a camera through the microscope and share our microscopy experience as far as regenerative soil goes and growing healthier plants. So yeah, we'll switch over to the microscope, and as soon as I popped on to this slide, we found a nematode. Stretching all over the place. He's probably a little freaked out from the light right now. Looks like he's got his tail kind of stuck on an aggregate. But anyway, that's a cool thing to find right when you first jump into a slide. I really haven't scanned around much. I was basically just getting centered up towards the top here, which is just up that way a little bit more. But we already found a nematode. All right, so before we dive into the microscope and scan around this slide and talk about what we see, we'll give you guys a little video tour of what we're actually looking at. So here we're coming into the garden. The first week of microscopy May, we checked out this. We got a bunch of nice, the walking onions are incredible. That we got from Garden Like a Viking. But yeah, we got a bunch of other starts. Sweet potatoes looking nice. Here's our cover crop that we actually left go to seed because I'm hoping to harvest these seed heads and use them next year for an overwinter cover crop. And then we got our wildflower bed over here. There's Lilu just hanging out, making sure the chipmunks don't eat my strawberries. Last week we checked out this strawberry bed, sorry. And yeah, strawberries are getting quite a few already, looking nice. This is the bed that we got on under the microscope right now. This is my sweet potato bed. I just started this bed last year, filled it mostly with Costa Maine, just raised garden bed mix. And other than that, mixed in a little bit of my local soil, my dad's local soil, and yeah. So we'll go back over to the microscope, and there's that nematode wiggling around. But that line of greens was a mix of, in the middle of that bed, was Simpson lettuce and also some arugula, some rocket arugula, both from Baker Creek Seeds. And I find growing greens in your sweet potato beds works out really nice because if you just only have a little bit of bug pressure, which we have a lot of bug pressure here in New England, but if you got nice healthy plants, you only get a little bit. And yeah, I find that my greens, which will get devastated all on their own in another bed away from other beneficials, will look perfect, and the bugs will only eat the sweet potato leaves and leave my lettuce alone. So that's how I keep my lettuce looking nice without a bunch of holes in it. It'll still grow fine. Like, I won't lose a crop from bug pressure. But anywho, back to this nematode squirming around. Gonna see if I can do a little bit of digital zoom. We're definitely not going to be able to tell what type this guy is because the way that he's moving. But I don't know if he's just stuck in there. We do see there's a fungal piece of fungal hyphae just popped out of that aggregate. But he doesn't seem to be stuck on fungal hyphae, but he might just be feeding like that. He might not actually be stuck. He might just have his tail wrapped around the aggregate and using that as an anchor to whip around and feed on stuff. But it's interesting how he's just spiking around at things. I would almost think that would be the predatory action of something that was looking for, a fungi or a root to stick its head into. I feel like the bacteria feeders, since they're all over the place, would just be more sort of, you see them, their heads, the ones that I've identified as bacteria feeders, just seem to be kind of slowly swarming around. But you never know if this guy's just stuck or freaked out because of we're blasting him with way more light than he's ever been exposed to. But yeah. Hi, just some night owl. Just some night owl. Good to see you here. Yeah, if you guys are joining in, feel free to drop a comment, ask any questions. Yeah, let me know where you're joining from, and what are you excited about growing this year? And yeah, let's see. What's this? Is that a super large? No, I think we're looking at a small root here. Yeah, that's too large to be a fungal hyphae. We are at 100x, and right now we're at 2x digital zoom magnification. So here, we'll go back out to, so there's stock 100x under the microscope. Yeah, from Estonia. That's pretty cool. That's just some night owl in from Estonia joining us. Glad to have you here. Yeah, so any questions about regenerative soil or microscopy, feel free to drop in the chat, or it's just kind of doing a fun microscopy day where we just throw some stuff, a different soil sample under the microscope every week and just hang out, chat, scan around, and you never know what we might find. So, all right, so right here in the middle, as far as what we're looking at, that is organic matter. Very broken down organic matter, but it's definitely different from, the minerals and bits of sand, silt, and clay. You can see, let me grab my pointer here. So you see, like, that's a bit of probably silt or clay, but this is all organic matter. Here's an aggregate. This might actually be a mineral that's just covered in glomalin that is looking so dark color like that. We can kind of go over here and just look a little bit closer. Yeah, let's actually, I got this polarizer here to see if, yeah, there's a little bit of seeing if any refracted light is coming in and off of that. So I'm just twisting a second polarizer on my light source. I'll give you a better example of when that comes into effect here in a little bit as we go through the slide. So yeah, I'm going into 2x here, or actually we'll go into 4x, and we see a very clear fungal hyphae. Look, it's going right in and out of that aggregate, connecting everything. So even though it's clear, it's very uniform. So it's probably a new growth. It's funny how fungal hyphae can look very different depending on its stage of growth. So it's tough to say that, just determined by a dark colored one to a clear colored one. Could be different species just based on its age. And yeah, so anyway, whole nice big cluster of aggregates here, which is just the soil forming that structure with the help of bacteria. So that's organic matter, minerals, and everything just all sort of clustered up. And that's all done by the biology forming glomalin, which is kind of like a glue, which helps them, pull the nutrients they want out of the sand, silt, and clay, and also the organic matter. So, yep, organic matter. Right here, we can see kind of the decaying, plant cells in between there, even at 100x. And it's probably a chunk of mineral stuck on top of it right there. So yeah, when we're going around, we're just observing kind of our, under the microscope to judge the health of our soil, just like we would witness the health of our plants. There's a lot of subtle things that we would notice. But what I'm already noticing here is just the overwhelming number of aggregates compared to the small little sand, silt, and clay. So, which is to be expected by, buying a nice, somewhat expensive. And Costa Maine is definitely one of the best beds that soils that I have microscoped compared to other bag soils that I have checked out. But yeah, you can see, so these big pieces here would be sand, and then smaller ones, silt, and then these little tiny pieces would be clay. So that's all clan, silt, and silt and clay is just their particle size. And they could be made up of a bunch of different stuff. Oh, this is kind of looking interesting. See that right in the middle? That doesn't... We'll zoom in a little bit. Very odd looking. Just the color and whatnot. I would say that would be an amoeba. But, yeah. I don't know if we could do anything with the light and the shading here to get a better look. Just opening and closing the aperture of the light. Maybe go up and down. Turn down the light, up the light. And then maybe we'll just, we'll kick on the epi light. So this is epi-florescence, part of the Lumen. And kind of turn down our backlight just to see if anything is glowing. Really got to turn it off and change our settings quite a bit, but I don't see anything glowing super bright in there. But this makes things with phosphorus in it phosphoresce. Oh, and I'm actually noticing, you see that little, he just went right under there. Where's my pointer? But a flagellate. Little bumbling. It's right over here. Let's see if I can get a focus on him. Oh, I think he went under the amoeba looking thing. The flagellates are very small, usually just one cilia. But yeah, even at 100x with our digital zoom, we're seeing the bacteria wiggle around there. We spot a flagellate. I think he's actually... Oh no. Alright. I lost my pointer. But anyway. We'll go back out to stock. We're going to do a little bit more scanning around. Oh, so epi-florescence is a LED light that is at a super narrow wavelength, which is pretty much cyan blue. There's a couple of microscopes, but LW Scientific is the only one that I know of that makes one. But I have heard of there is another company, but it's just this module that goes on the top of the scope here. Yeah. just right here. And so when I turn on this light, it puts that bright blue light onto the scope from above, and it makes things with phosphorus glow and phosphorize, which I always find is funny as far as, like luminescence goes, and phosphorization. Phosphorus is when phosphorus glows, but scientifically, like, luminescence is just basically another way of saying there's light coming from something that we can't understand or explain. So it's pretty interesting. It's like we basically, understand the chemical reaction in fireflies and stuff like that glow and produce light, but we not quite fully if you look into it. I wouldn't say we fully understand it. If you do the math on what it should be, it doesn't quite add up. I think they're brighter than it should be. But anyway, I think we see an amoeba right there in the middle. Yep, small amoeba. Oh, see if I can get a little bit better focus on them. Yeah, there we go. Yep, small amoeba. And then, where's the amoeba? I got a bigger view over here. And then we got a fungal hyphae. So you can see, and a fungal spore too. Wish I didn't mess up my pointer. Here, there we go. I fix it. Now we got a pointer. So fungal spore is right there. This is the fungal hyphae. You can see the septi separation in the cells. Got another fungal hyphae over here and another one out of focus. Yeah, I can adjust my focus a little bit, see if we can get them. Yeah. Good, beneficial, most likely. Saprophytic fungi, breaking things down. So yeah, we'll go back out to stock. And just kind of scan around. Yeah, if you're just joining, feel free to ask any questions. Say where you're joining from. And yeah, what are you excited about growing this year? Let's see, I think we haven't gone much over to the right. We kind of want to scan over here. Let's see if we can find anything cool. Oh yeah, look at how that aggregate's all connected together. Large stuff. It might be roots connecting all that together. Let's see what else we can see. Eventually here we'll click over and show you the full potential of the epi-florescence once we find a nice root. Alright, we scanned all the way over to the full right of the slide. We'll go back up. So far as big microorganisms, we found one. But if you think that I'm just putting one drop in the slide that's diluted by 10 by water, and there's 75,000 estimated drops in a gallon, even if we see just a couple of big microorganisms, like just do the math and we're into the low millions pretty quick. In just 1 gallon. Just from seeing one on there. And the amount of fragilates and bacteria that's in there, the numbers are just pretty astounding. I did see, it could have been a mineral. The polarizer does make it blue, but if we do see usually plastic under the microscope will look like a hair. It can almost look like a really odd, like right here, that's some sort of mineral actually glowing blue. So if I take the polarizer off, it doesn't glow. But we put it back on and could do some interesting stuff with the different minerals and stuff like that. And this is just using a polarizer. So we want it right there where it's pretty stock. So it's just enhancing our color and blocking blur, giving us a sharper image. Because we don't want it affecting the color too much. But like I always like to think of cobalt as being blue, but who knows what it actually is. But it could be a little piece of something. It's some type of mineral that is just refracting light. Yeah, it's pretty wild what a polarizer can do. It bends all that refracted light, like just like hanging one of those gemstones and making rainbows. But it also helps tighten up your image and stop a lot of that light bleeding around so you can see stuff a lot clearer. But I really want to experiment with some different minerals and stuff to see if there's any correlation between the different way things bend light under the microscope and with the polarizer. Because I've only really found it to do this when I twist it like this that it goes dark. That only happens when you have two opposite polarizers. So I have one in the camera and also one under my light source. And right now I'm just rotating the one under my light source. So it's kind of fun to play with. All right, so let's see. I think we've scanned around quite a bit. There's some nice big chunks of organic matter. Wonder where that nematode went, but here let's actually we're gonna switch our objectives into 400x now, so we can get a little bit closer, see some bacteria. Maybe we pick up on some smaller microorganisms that we missed in our scan. Even though we didn't do a very scientific scan, I kind of just went willy-nilly all over the place. But we need to adjust our settings on the camera. So we're just changed our shutter speed. Yeah. So now we're gonna... So we are at stock 400x. Dialing in my focus just a little bit. Oh yeah, there's quite a bit of bacteria. Yeah, we'll zoom in. Yeah, we'll go actually 4x digital zoom just so you guys can see as close as we can in case you're on a smaller screen. But so we got bacillus shape right there. Around one are cocci. And those are the two main shapes morphologically that we see in the soil. Yeast are usually a little bit larger and round, and they don't usually wiggle or move too much. But if you get a good focus on them, you can kind of see their cell walls. But yeah. Oh, there's a quick moving one. Where'd he go? Oh. So every once in a while you can see a fast moving flagellate, which are just a little bit bigger than the large bacteria. Oh. Let's see. Got a very round looking thing. Yeah. That's an amoeba. Or stuff. Sometimes fungal spores, especially when they're this small. Like we're at 4x digital zoom here. If we go out to 1x, yeah, that could be a Oh no, we are actually looking at this right here. Yeah. Could be a cystin organism or possibly a spore. Most of the amoebas you see have a little bit more shape and whatnot to them. I'm sure we'll find one here eventually. Got a couple more nice pieces of fungal hyphae. I'll do a quick zoom in on them. Nope. Nice brown colored fungal hyphae there. And one more over here. Just a little piece. Let's see. This might be a root right here. Yeah, it might be just a little. I think that's a little segment of a root. Looks pretty fresh. So let's throw on the epi light. Turn off our backlight. Got to change our settings just a little bit here. Yeah, let's go up to, yeah, right about there. And now actually we're going to turn the bottom light off completely. So now we are just illuminating from the top, epi-florescence light. And looking at this, yeah, seeing how it's so orange like that. I think this is, hasn't been living recently. See the glowing little bits there. There's where some phosphorus is. But most live roots look a lot brighter than this. But yeah, just another way of looking at things. And oh, so we see a, come on, right there. Yep, so that little tiny thing there is probably just a little piece of phosphorus of one form or another. Or something with a lot of phosphorus in it. You know, some of, that little piece right there might have been fungal hyphae, might have been some sort of organism with phosphorus in it, but yeah, pretty much everything under the super narrow wavelength that has phosphorus in it glows bright green. And then other things glow like the oranges and the reds, but they're not specific. Like sometimes when you see this oranges glow, I think it can be like calcium, but it could be other things. But when you see the really bright glowing green, that's when you know it's phosphorus. But it's cool how, yep, so that up there. Yep, organic matter. Oh, yeah, you can definitely see, yeah, right there is a perfect example. So right here is, that's all in between the cells of this piece of organic matter that's glowing. And the reason why it's glowing is because there is endophytic fungi growing through there and pumping phosphorus and exchanging the plant for its sugars and exudates. Take a look at this. Yep, you can see more. So here we're seeing just more of an orange. So that's just the calcium of the cell walls. We don't see any bright green in there. We just see normal orange and it glowing. So you can make out the edges of the cell wall, but no phosphorescence. Which is probably because this looks more to me like a a little piece of a leaf or something by the shape of its cell walls that's just being broken down in there. It's not a living plant, so it doesn't have that symbiotic relationship and that trade and that inoculation of the phosphorus. We can almost see little, right there. So we can even see like old strands, and you can see fungal hyphae that was growing inside of this piece of organic matter, possibly at one time. But right now, it's not funneling and exchanging phosphorus, so it's not super bright green. But yeah. Another piece of organic matter and everything. It's got all those little pieces of the clay that are glowing have a little bit of phosphorus in them. Oh, what do we got over here? Oh, this is interesting looking. That's definitely something growing and living in there. Very odd shape. And quite large. Yeah, we're at 400x. Huh. Yeah. So you see, this organic matter has a very interesting shape. But it's still not... It's glowing a little bit, but not as bright as, say, this over here. Yeah, so this would be an example of just organic matter with a little bit of phosphorus in its cell walls, but still not mycorrhizae. You've seen some of my other images where the roots are growing super bright. Posted a couple on Instagram and whatnot. This is glowing bright yellow, not quite bright green. Not phosphorizing. Got some bright things over here. Yep, that looks like mineral phosphorus. But anyway, yeah, it's pretty neat. Looking around in epi-florescence light. That looks like an amoeba right there. But yeah, we'll click off that and turn back on our bottom light. Turn up our brightness. Oh, wow. Yeah, I would have passed right over that. And under epi light, it actually looked like a living thing. But I don't know. It's very, very dark brown. So it's probably broken down. But yeah, right there in the middle. That was interesting how we saw it under epi light. And you could definitely see that it was something living at some point. But now it's all dark brown, and I think that's because it's probably was an amoeba, but now it's a dead amoeba and breaking down, and those amino acids and glomulins are being formulated around it, which kind of like a rust, turns everything brown. But it's actually the opposite of rust, because this is reduction, not oxidization. Oh, yeah. Got another very round thing right there. Could be an arcella or a large spore. Light that's kind of bouncing off that piece of sand up there. Couldn't see it very well. Yeah, it's always interesting when you see these blue things. I always like how the light changes. Just as you twist the polarizer. It's pretty interesting. Refracted light is neat. basically bouncing. I'm not even sure if I'm using refracted light. I'm not a light expert, but just when it bounces and goes through prisms and just like that little crystal puts rainbows on your walls when you hang it in your window. But there's a reason for it. It's not random. So I wonder if we can correlate it. Large piece of fungal hyphae. Looks like it's breaking down. It's got bacteria inside of it. Yeah. It's a pretty wild world. And all these microorganisms are communicating, sharing DNA, doing horizontal gene transfer, which if you don't know what that is, it's pretty simple. It's just there's vertical and there's horizontal. So vertical is like us and mammals. The parents get together and they pass their genes and their DNA down onto their offspring. But horizontal is basically two bacteria come into contact with each other, don't necessarily even need to reproduce, and can share a gene or a trait or a little bit of DNA. And yeah, we're just learning about how that happens way more than we thought. See a little football-shaped fungal spore in the middle. So I always like to think, and if you just think of if that's happening all the time as far as environment and what we know from how much things can change, I just think of, the bacteria down there and going around in a healthy soil environment where there's plants pumping down root exudates and everything like that. When a pathogen comes along and says, hey, I got this DNA trait that lets you eat living cells. And that particular E. coli is like, nah, bro, I'm all set. I got plenty of plant exudates and plenty of food. I'm good. But then take a big, ecological disturbance, and now all of a sudden there's no plants and no other nutrient cycling going on. That same pathogen comes around to that same, perfectly harmless form of E. coli. And it's just like, hey, I got this DNA trait that lets you eat living cells. And yeah. But anyway, we got some nice fungal hyphae there. Oh yeah, so that's a perfect example of, nice large fungal hyphae, and you can see the bring the pointer out, so see the separations, the septi in the cell wall. It's nice and uniform. Slight brown color by its size, most likely a saprophytic fungi, which is a great reducer. Reducing organic matter, reducing wood matter, can also mine minerals, and cycles nutrients throughout the soil. And is just an overall extension of your plant roots too. Well, more endophytic fungi, but even the endophytes, like the saprophytes, being outside the root, endophytic meaning inside the root. Like the mycorrhizae. We're seeing there's a flagellate moving slowly. We actually see there's one right up above. Something bubbling around in here, and then the slow moving one just went right over here. Yeah, there it is. It's either looks like a flagellate. Could be a very small ciliate. Just because the flagellates are usually bumbling about, just like this one that we can't really see very well because it's stuck in there. Oh, anyway, another example is I think that's definitely a cyst right there. There is. Let's go into 4x. Can't get a great focus on it, but this thing's perfectly around, and that might be a nematode inside a little egg. Having trouble with the focus, but looked more like a cyst from the outside. It's also a very round, faint thing right there. But I definitely think that's something in a little shell. Anyway. Yeah, make sure I'm not missing any chat. No, I think that's about it. But all right, what do we got for time? Yeah, we've already done a pretty good scan around. Let's see, was there anything else I wanted to do? Maybe we'll look at this with the polarizing effect. Oh yeah, that makes some a little bit of cool colors. That's pretty neat. And that's a mineral with some organic matter on it. Maybe even some fungi. There's a bunch of stuff growing on there. Trying to pull those nutrients out. We see a bacteria wiggling around on there. Probably extracting something. Yep. Microcosmos can look pretty cool. Yeah, we're at 400x, 4x digital zoom. We'll go back out to, so here's stock 400x. Yeah, but I don't know. I think we might wrap things up. See some more fungal hyphae. Yeah, but overall, I'm really happy with the amount of aggregates. This is a brand new bed. We got great levels of organic matter. There's definitely in the coastal main soil. There's definitely plenty of organic matter in there. Nice mix as far as those ratios go. And yeah. Plus the local soil that I mixed in with it. Plus a little bit of my own compost. Seemed to build it up pretty well. We're definitely seeing more stuff like this than stuff like this, which is the sand, silt, and clay. That just gives you an idea of what your organic matter level is under the microscope. And then you see giant things like this. We can focus a little bit here. Yep, that's definitely, any sand, silt, or clay that big you wouldn't be able to see through. We can't quite see the plant cells anymore because this is already very pre-digested. And we're just seeing looking at a colony of bacteria on there, breaking that thing down. This almost looks like just how square that is. Usually anytime you see something square, it's a diatom. But that might just be a fragment of something or other that's square. Definitely can see some bacteria on it, but so some life is interested in it. But yep, nice big aggregates like this. And the fungal hyphae connecting everything get together, forming that sponge. And then after that, nice healthy amount of bacteria, some amoebas. I'm pretty sure that's an amoeba right there. Might be a round aggregate. Let's go in a little closer. No, I definitely think that's an amoeba. Maybe. Yeah. Yep. I go 98%. That's an amoeba. Very round, very oval-like. It's interesting. We've seen a lot of those weird shaped ones. I haven't seen we did a pretty good amount of scanning around. Definitely didn't do a very good scientific job getting every corner and every spot of the slide. You kind of want to mow the lawn, go up and down, and I kind of like my lawn would look like there'd be a bunch of missed patches on how I went around this slide. But anyway, yeah, that might wrap it up for this one. Unless anybody has any last questions, please feel free to drop any questions about regenerative soil or microscopy, soil microscopy, into the comments, and I will do my best to answer. And saw something moving under there. Yeah, it's a very interesting grid-like patterns and round stuff. Oh yeah, there's definitely I think that was a flagellate circling around underneath that piece of organic matter. Hey, thanks, Old Kiefer, for joining. You're very welcome. He just said thanks for sharing. Glad you could join on. But yeah. Alright. I think that mineral is going to look cool under polarization. Let's check it out. I got another just blue one. Alright. Well, yeah, I think that's going to wrap it up for this stream of my strawberry bed. Oh no, this was the sweet potato bed. Strike that. But anyway, yeah, this was, again, this bed was just made last year, mostly coast of Maine soil. And yeah, pretty happy with it. Definitely with the organic matter levels, the bacteria levels are looking really good. Lettuce seems to be taking off. My sweet potato starts seem to be going really good. And I'm stoked that I got that purple sweet potato, which didn't do well last year at all. And all of a sudden it just popped up and it's the best looking one. So yeah. Anyway, gonna be posting a garden tour soon, so subscribe and hit those buttons if you would be so kind. And I'm gonna put some videos up there and whatnot, and some buttons for you guys to click with the end screens. And until the next one, we'll catch you on the flip side.